Flow cytometry. In flow cytometry, the sample cells have fluorescence molecules on them. When the sample is inserted, it runs through the nozzle. The nozzle has two parts: the central core, where the sample is initially inserted, and the outer sheath, which contains fast-flowing fluid. The fluid in outer sheath forces the sample cells to move in a straight line, one by one. This phenomenon is generally known as the hydrodynamic focusing of sample cells. Now a laser light is thrown on moving cells. When the light strikes the cell, two events occur. Light scatters and fluorescence occurs. The light scattering occurs in two directions. Therefore, two distinct detectors are used to detect these fluorescence signals: the forward scatter detector and the side scatter detector. The forward scatter detector is placed in front of the laser light or parallel to it, while the side scatter detector is mostly placed on the side of laser light or perpendicular to it. The side scattering of light is mainly caused by granularity and structural complexity of the cell. Thus, a cell with a more granular structure will cause greater side scattering, while a cell with less granular structure will cause greater forward scattering. The cell causes fluorescence when the laser beam hits the cell. This is because the cell absorbs energy from the laser light. The fluorescent molecules on the cell start glowing. This way, the cell emits the absorbed energy by causing fluorescence. The detector detects this fluorescence and stores it on a computer. Flow cytometry could be used to separate cells of interest from a complex mixture. This is the colossal application of flow cytometry. Suppose we have a mixture of cells with red and green fluorescence molecules. We want to separate the red fluorescence cells from the mixture. Hydrodynamic focusing forces the cells move in a single line one by one. But this line breaks into regularly spaced droplets. Each droplet contains a cell in it. To separate the cells, an electric field is applied by placing two plates around the droplets. These plates are electrically charged and are called deflection plates. The deflection plates apply electric field. When the droplets move through these plates, they experience the electric field. So they get deflected towards oppositely charged plates and drop into different tubes. This way, the cells are finally separated and collected into different tubes. The black droplets have no charge on them, so they do not experience the electric field and fall into the middle tube. This process is known as cell sorting or fluorescence-activated cell sorting. The word cyto means cell and metry means to count or measure. So flow cytometry is a powerful technique to count and measure various physical and chemical properties of the cell.
That is it for today's video. Make sure you are subscribed to the Science Info channel. Like and share this video if you wanna help others. Stay tuned. I will see you in the next video.